exciting to be here, and thank you for having us.
Şu anda sessizleri görün.
I want
Tamamdır.
to talk about our research, which we've been conducting for almost over 10 years on propolis and breast cancer. My name is Alison Pınar Aranat. I've been working with Professor Oğuz Öztürk in Istanbul University. Okay. I want to first start with breast cancer. It's the most diagnosed cancer in women, and annually 2.3 million women are diagnosed with breast cancer. And also, depending on whether it's a developed or developing country, it's also the leading cause of cancer-related deaths in women. So it's really significant. And among all other cancers, in 2020, the 25% was just breast cancer. And it's estimated that one of 8 women will sometime have breast cancer. So throughout years, of course, screening reduced mortality. It enabled us to see the precancers and remove the precancerous lesions, or treat in early stages, which means that they're usually more treatable. But there are different types of breast cancer, and the aggressive types, when we're talking about the aggressive types, the overall survival is really low, and response to therapy is also low. So when we said breast cancer can also be seen in younger women. In the US, 10,000 women diagnosed every year are under 40. In Asia, 13% of all breast cancer cases, the patients are under 40, 5% under 35. And in Europe, this number is also 5% is under 35. The most important part here, usually the breast cancer diagnosed in younger women, they're more the aggressive type, and the response is lower. Let me talk about shortly the different types of breast cancer here. The first type, the most common type, which comprises the 50 to 70% of all breast cancers in women, the hormone responsive type. What makes it hormone responsive? They overexpress estrogen and progesterone receptors. Here, the main treatment approach is estrogen blockers, usually endocrine treatment. They block estrogen that stimulates tumor cells to grow. Hormone responsive types, they're associated with better prognosis. But there are also more aggressive types. For example, HER2 positive type. Here, the human epidermal growth factor receptor, HER2 receptor, is overexpressed in this type. It's highly aggressive, and it comprises of 20% of all breast cancer types. It's associated with poor prognosis, and it has high recurrence rates. The patients here are usually, most of the patients diagnosed with HER2 positive breast cancer type are under 45. Here, there is a targeted therapy. It's called trastuzumab. It's a HER2 antibody. But the main problem here, the 2 of every 3 patients is already resistant. And the remaining, like 30% or 1/3, the patients, they acquire resistance. They become resistant after a while. So the treatment is very limited. And response to treatment is not as much as we want. The third type, we can say, the triple negative type. It is the most aggressive type of breast cancer, And because it's not expressing any of these estrogen, progesterone, or HER2 receptors, that's why it's called triple negative. It's the most aggressive type, and there is no targeted therapy. So we need to use conventional methods here. Its recurrence rates are really high. And again, 50% of all breast cancers are triple negative breast cancers, and most of the patients here are under 45. And most of the time, a lot of patients, they do not respond to chemotherapy. So we need a new solution, a more better, and with a solution with almost no adverse effects that will improve the treatment in these cancers. So the conventional cancer treatments, it's comprised of agents that usually induce DNA damage or cycle arrest or induce apoptosis. These are the chemotherapy agents usually comprise these kind of agents that induce these kind of damages. So, but the main problem, cancer cells, they can develop drug resistance after a while, and they work through different cellular mechanisms. They can also, throughout therapy, they can change their type, because if you block the certain cellular pathway, they can just switch to another pathway, which is commonly observed. So it's important to target multiple cellular pathways at the same time to overcome this problem, to just effectively kill these cancer cells. And also there's another point, it's the intertumoral heterogeneity, which means in a certain tumor mass, there can be different types of breast cancer cells. So your treatment approach may affect some of them and may not affect the rest, which means cancer cell will recur. So the main thing here, this researchers now are talking about this new concept, confusing complex system with many weak hits. Therefore, we're going to be able to target multiple cellular pathways at the same time. So how can we confuse this complex system with many weak hits? This research, then researchers started to look at propolis. Propolis, owing to its content, it contains sometimes over 300 different compounds were identified in different samples, propolis samples. So this many hits with many weak hits means different compounds that being together with low doses. So due to its polyphenolic content, researchers turn to propolis. So when we talk about polyphenols, they usually individually, when you administer them individually, you need, because of their low bioavailability, you need to administer them in high doses. But when you use them in combination, you can use them in lower doses because of their synergistic effects. So it increases when they're together, they're much more effective. And they have all these different effects on anti-carcinogenic, neuroprotective, cardiovascular protective, and all that. So the main thing we can talk about their antioxidant properties. So we can summarize it with like free radical scavenging effect. They can capture heavy metals. They can inhibit different types of oxidases and stimulate certain antioxidant enzymes. They also have pro-oxidant mechanisms, which with formation of reactive oxygen radicals and then causing oxidative DNA damage, they induce mitochondrial dysfunction, which is associated with apoptotic cell death, which is what we want to induce in cancer treatment. So different propolis samples have different content because of the environment, because the bees collect all these polyphenols, all these compounds from the environment. So the propolis samples we collect from different regions can have different contents. So the most common type, the Anatolian propolis, or the propolis collected in Europe and North America, in some regions of Asia, they're poplar type propolis. And also there are different types, like Brazilian propolis. It's they all have different bioactive contents. The Brazilian propolis like green, and Anatolian propolis is like brownish. So it's all depending on the bioactive content. So how you extract these different propolis samples also contribute to the content. So many researchers use ethanol to get better results, to enable to solve more compounds. In our previous study, we evaluated different propolis samples on different breast cancer cell lines. You can take a look at it if you like. In this study, we evaluated 7 samples. And as you can see, all these samples had different compounds with different concentrations. So in our study, you can further take a look at the paper. We obtained the best results with this one propolis type. Here, the distinction here, this propolis type, it contains a more diverse range of compounds compared to others. So maybe lower concentration sometimes, but the main thing here is the diverse range of content. So we confirmed that this propolis sample was inducing apoptosis in these different cells. And also we, in order to confirm that it's only effective on cancer cells and it's safe for healthy cells, we also administered it on fibroblast and fibrocystic breast epithelium. And our results show that it was safe. So further on, we wanted to see the molecular pathways that this propolis sample is working on. So in different hours, we evaluated the genetic alterations. On the 24th hour, 191 genes were upregulated, 441 was downregulated, and the 48th hour seems more effective. This number went up to 456, and downregulated genes were 1,090. So when we evaluated the many pathways were affected by propolis, but in general, we can talk about when we talk about the 48th hour, the downregulated genes were members of cell cycle related pathways and interferon signaling pathways, and also estrogen-dependent gene expression. And upregulated apoptosis-related pathways, and also response to organic substances, and signal regulation, which is important and tells us what this is what we wanted to see. And also we want to, as I said, we want to target different types of breast cancers at the same time. We want this to be able to be effective on different types. So on this other cell type, this one was representing the hormone responsive. This cell line is representing the HER2 positive type. Again, we have different numbers of genes altered here. And again, we can talk about here, immune response was an upregulated group, which is the apoptotic effect may be associated with it at the same time. And also here, the downregulated pathways, genes were members of cell cycle, DNA repair, cellular response to stress, and DNA damage response. So since these genes are downregulated, cells are not able to go on with, continue to divide or repair their DNA. And again, the third type cell line was representing the triple negative cancer, breast cancer. So again, here the 72nd hour was more genes were altered there. The upregulated genes were more of the immune response related pathways, and downregulated are cell cycle related pathways. So here, I'm over time, I think, and here we observe that propolis, this propolis sample with high diverse range of polyphenol compounds induce the cellular pathways as we want them, as we prefer. So bottom line, different propolis samples may have different effects depending on the content, and these samples must be analyzed for their content because due to the environment, you can see like heavy metal residues or pesticides. So you have to make sure you don't have these unwanted residues. And then bright propolis can be promising in breast cancer treatment. Thank you for your attention.
Dear participants, first of all, I would like to thank the organizing committee, especially dear Dr. Ali Timuçin Atayoğlu, for the kind invitation. I am a medical oncologist and also have a master's degree on molecular biology and genetics. It's very pleasant and nice feeling to be on this platform because cancer is still an important health problem all over the world. Despite rapid advance in treatment options, it still ranks first among cause of that. We will learn more later as we understand the molecular biology of cancer. This is very important because every new molecule, every new drug and new research will be promising for cancer patients. Therefore, apitherapy is an important and promising research fight for cancer. In order to express more clearly as a clinical oncologist, I will continue in Turkish in comments and questions section, and I apologize in advance. Now we will listen to the scientific evidence about propolis in cancer by Professor Erdem Yeşilada from Yeditepe University, Istanbul. You can start for presentation, Professor.
Thank you, Professor Tanrıverdi. First, I'd like to congratulate the organizing committee, particularly Dr. Atayoğlu, for their efforts on organizing the first International Apitherapy Congress. This would also help to draw the attention of the worldwide health providers, physicians, and the scientists to the benefits of bee products in medical practice, medical care. Yes. There's an old German saying, beekeepers never get cancer. Only 1 in 3,000 beekeepers suffer from cancer, while 1 in 3 non-beekeeping people get cancer during their lifetimes. This means that beekeepers are 1,000 times more resistant to cancer than are ordinary people. That is what the question asked by Desider Grunderberg. Have royal honey, royal jelly, and propolis. These are the main 3 major bee products. But the first 2, there's no significant anti-cancer properties. How about has propolis as anti-cancer properties? From Columbia University, Professor Grunberger asked himself this question, and around the mid of, this was around the mid of 1980s. Then, in collaboration with Professor Nakanishi, he was a very famous, eminent organic chemist from the same university. They started testing anti-cancer properties of an extract of propolis from Israel. They obtained from Israel and found that a compound called caffeic acid phenethylester, CAPE, in ethyl extract, ethanol extract, kills cancer cells without affecting the growth of normal cells. Then this, I think this is the most likely that propolis makes beekeepers so resistant to cancer. When I search in PubMed to find the published investigations on the medicinal features of medicinal characteristics of propolis, I found 3,645 published papers, articles, mainly after the year 2000, as shown here. And among these, 1,010 were published on the subject related with cancer. This means that about 1/3, almost 28%. So this cancer is one of the main interest area for propolis to be used in medical practice. As shown here, when we compare the factors inducing cancer and the pharmacological properties of propolis, we can clearly observe that there is an evident counter-interaction between them. As you can see, free radicals and reactive oxygen species due to cigarette, alcohol, obesity, and other chronic infections and others. So propolis has a capability of powerful antioxidant activity, scavenge the free radicals. Chronic infections, in case of cancer induction factors, among the cancer induction factors, so bacterial, viral, parasitic, and others, it has a good antimicrobial characteristic. And weak immune system also another factor in cancer induction, also support immune system. Has a good anti-inflammatory activity against chronic inflammatory conditions inducing cancer. Also, for example, a good wound healing activity, and also maybe possibly active as prophylactic in genetic predisposition and other unknown factors of cancer. This figure summarizes the pharmacological effects of propolis on cancer cells. As you can see, reduce the uncontrolled growth of cancer cells, increase selective increasing apoptosis of cancer cells, reduce angiogenesis, regulates tumor microenvironment, interfering the specific calcium signaling pathway, hindering metastasis, and also support immune system, the body's own immune system. As shown here, we can classify the possible contributions of propolis under three main topics, main titles: on cancer cells, inhibition of uncontrolled growth of its anti-inflammatory antioxidant activities by it is antioxidant anti-inflammatory activities. Through this way, provide apoptotic effect, hinders the specific cancer signalization pathway, as also act as anti-angiogenic and anti-metastatic. On the other hand, also support the immune system, provides support of body's own protection system. And also, this is, to my opinion, this is one of the most important points, adjuvant effect. Increase the efficiency of chemotherapy or radiotherapy treatments. For example, increasing the sensibility of cancer cells to therapeutic agents, reducing the risk for organ damages, and also regulates the blood biochemical parameters, keep homeostasis of the blood parameters, I mean. The anti-tumor activity mechanism of propolis and its main active ingredient, CAPE, is complex and multi-directional as shown here. These are the factors stimulating the apoptosis, and these are the factors inhibiting the apoptosis. And CAPE act through antioxidant activity, reduce tumor induction, through its apoptotic activity, reduce apoptosis, and also through its action on inhibitory action on xanthine oxidase also, and also 5-lipoxygenase activity also reduce the inflammatory cytokines induction. Now, we have better say, give some examples on the effects of such effects of propolis and its active ingredient. First, inhibition of uncontrolled growth of cancer cells by its anti-inflammatory and antioxidant effect. As you all know that oxidative damage and inflammation, resulted inflammation, are the triggering factors in cancer. And propolis inhibits the release of kinases as shown here, inhibitory kappa B kinase and mitogen activated protein kinase. And through this way, provides inhibition of transcription factors and NF kappa B, cytokines, and others, and which will reduce, this will reduce the inflammatory pathway and of course reduce the cancer risk. On the other hand, for example, CAPE and some other phenolic esters inside the propolis with potent antioxidant activity reduce the reactive oxygen species damages by inhibiting the DNA synthesis of the cancer cells, I mean break DNA sequencing, also protein fragmentation and inhibition of membrane lipid peroxidase. Another point, targeting propolis targeting apoptosis of cancer cells. The other important point that propolis would involve cancer treatment in this case, and propolis, the important point, propolis provides selective apoptosis of cancer cells without any damage to healthy cells. And CAPE induces apoptosis by triggering the trail pathway, I mean TNF, tumor necrosis factor related apoptosis inducing ligand, and also another component of propolis, flavonoid chrysin, also activates caspase 3, and through this both three ways induce apoptosis, selective cancer cell apoptosis. Other points hindering the specific cancer-inducing signalization pathway. First, regulation of the tumor microenvironment. Second, hindering tumor angiogenesis and prevention of metastasis. First, tumor microenvironment regulation. The tumor microenvironment is the main environment around the tumor, including the surrounding blood vessels, immune cells, fibroblasts, signaling molecules, and the extracellular matrix. The tumor and the surrounding environment are closely interacting constantly here. And tumors can influence the microenvironment by releasing extracellular signals, promoting tumor angiogenesis, and inducing peripheral immune tolerance. So the immune cells can affect the growth or evolution of cancer cells. For example, black poplar type propolis, I mean, this is one of the richest sources of kappa, in 50 to 150 milligram per kilogram. And it is polyphenolic contents such as kappa and caffeic acid and quercetin, a flavonoid, reduce the number of tumors in lung cancer. The second propolis hinders tumor angiogenesis. This means there's anti-angiogenic effect. Formation of new blood vessels here supply nutrient and oxygen to cancer cells, gateway to the expansion of cancer cells and the formation of metastasis to other tissue organs. It has been suggested that kappa, by blocking this pathway, this signaling pathway, triggered by the NF-kappa B transcription factor, inhibits angiogenesis and, of course, carcinogenicity in various types of cancer cells by suppressing vascular endothelial growth factor. Vascular VEGF is secreted by the tumor cells and a strong angiogenic factor in the angiogenesis pathology, as shown here. Formation of new capillaries from the pre-existing blood vessels in cancer tissues, VEGF and its receptor are required. And increases tumor growth. This will increase the tumor growth, migration, permeability, and proliferation of endothelial cells, and contributes the metastasis of cancer cells. And it has been shown that kappa inhibits vascular endothelial growth factor receptor 2 signal pathway by suppressing its phosphorylation in endothelial cells. Another point, kappa is a PAC1 blocker. 70% of prostate and breast cancer, as well as the neurofibromatosis-related tumors requires PAC1. This is as shown here, serine threonine P21 protein activated kinase 1. And kappa blocks the PAC1 signalization to inhibit signalization. This is data in vivo animal experiment data as shown here. The third one under this section is anti-metastatic effect. Tumor metastases include extracellular matrix, ECM, degradation, which is regulated by matrix metalloproteinases. And matrix metalloproteinases are zinc-associated endoperoxidase enzyme and have a role in cancer proliferation by extracellular matrix components. The matrix metalloproteinase gene is expressed in response to stimulation by several factors here, as shown here. Some, for example, cytokines such as TNF, interleukin 1 beta, interleukin 6. These are pro-inflammatory. These are inflammatory cytokines. And matrix metalloproteinase activity is regulated by several types of inhibitors, among which TIMP, tissue inhibitors of metalloproteinases. This is the most important one. And the balance between matrix metalloproteinase and TIMP is important. This is the responsible factor for controlling ECM protein degradation. And CAPE is a potent inhibitor of matrix metalloproteinase 9. Matrix metalloproteinase disrupts the collagen type 4, which constitutes the main membrane and ECM structure and provoke the cancer metastasis. That's why the action of CAPE is important. And another important point is anti-metastatic effect of propolis is higher than the individual components of its other phenolic compounds. The other point is regulation of immune system, immune modulation. Propolis increases CD4 to CD8 T cells rate and number of TH cells. And CD8 T cells are also known as killer T cells, and these are cytotoxic. They are able to directly kill cancer cells. And CD8 T cells are also able to use cytokines to recruit other types of cells when mounting an immune system. And activated T lymphocytes through this one stimulate the cellular immune response and eradicate the malign tumor cells. Propolis stimulates the activity of CD8 T cells, interleukin 2 and interferon, and also nuclear factor, natural killer cells. And in advanced melanoma cases, combined use with chemotherapy of propolis may provide additional benefits. Here, for example, normal and stress-applied mice, propolis did not induce remarkable increase in interleukin 2 level, while after melanoma induction, interleukin 2 production reduced. And propolis significantly increases TH1 cytokines, interleukin 2 and interferon gamma. CAPE also increased the lytic effect of natural killer cells on tumor cells. Specifically, this is important. T and B lymphocytes exert their effects on cancer cells only after their uncontrolled growth. However, NK cells exert their effects immediately after finding the target cells. Killer cells, together with adaptive immune system, trigger the cytokines responsible for regulation of T cells. And NK cells are interacted with interferons, interferon alpha, beta, gamma, also transaction growth TGF, beta 1, and some important interleukins, as shown here. So this means that CAPE pro-apoptotically affects the only cancer cells without affecting the healthy cells. This is important. And also another, CAPE also triggers apoptosis by caspase pathway. As shown here in the figure, stop cell division at phase G2 to M and stop cell cycle at G0 to G1 by inhibiting the phosphorylation. So CAPE selectively provides apoptosis of cancer cells while does not harm to normal fibroblasts. Another point, one of the most important to my opinion, adjuvant effect of propolis. CAPE enhances the effect of chemo and radiotherapy on cancer cells. CAPE, by inhibition of glutathione S-transferase enzyme activity, reduce the glutathione level and increase the susceptibility of tumor cells to chemo or radiotherapy applications. So reduce the efflux pumps, you see, effect of efflux pumps. And the other point is CAPE also reduces the inflammatory reactions induced by NF-kappa B cascade, which is triggered by radiotherapy, as shown here. After the radiotherapy or chemotherapy induction, inhibitory kappa B disappeared and release through this way, NF-kappa B release. And release NF-kappa B induce anti-apoptosis survival and also proliferation of inflammatory cytokines, induce tumor promotion, induce metastasis, induce angiogenesis, induce inflammation, and also induce immortality of cancer cells through telomerase enzyme. And these factors also involved in many of the cancer types. This figure shows the adjuvant effect of CAPE in several ways and has the sensibility of cancer cells to chemotherapeutic agents. Multi-drug resistant one gene in cancer cells increases resistance to chemotherapeutic agents. And CAPE downregulates MDR1 gene by reducing the efficacy and number of related receptors. Also, CAPE reduces the defense mechanisms of cancer cells to chemotherapeutic agents, particularly 4-hydroxy-2-nonenal. HNE is a highly reactive protein-bound aldehyde formed as a result of peroxidation by reactive oxygen species of membrane lipids. And CAPE inhibits HNE detoxifying enzymes expressed highly by the gastric cancer cells, leading to accumulation of HNE. Another point, CAPE may have additive inhibition of cancer cell proliferation with chemotherapeutic agents. Incorporation of CAPE into, for example, fluorouracil may reduce the amount of fluorouracil requirements and of course reduce the cost of therapy and of course protect from the severe effects of this chemotherapeutic agent such as diarrhea, nausea, vomiting, mouth ulceration, appetite loss, watery eyes, photophobia, metallic taste, impaired blood parameters. Another point, CAPE disrupts the endogenous antioxidant system in cancer cells by acting selectively on tumor cells without affecting healthy cells. And combined use of CAPE with chemotherapeutics may provide selective direction of chemotherapeutic to cancer cells. This is also an important point. Regulation of the impaired blood biochemical parameters by chemo or radiotherapy also another characteristic of propolis. Propolis may prevent risk of infectious diseases in cancer patients due to reduced immunity and leukopenia induced by chemotherapy or radiotherapy. For example, Brazilian propolis prevented leukopenia and reduction in immunity induced by fluorouracil and other chemotherapeutic agents. Either aqueous or ethanolic extracts of propolis acts as radioprotective in acute gamma ray treated mice, prevent DNA damage, and also provide reduction in blood leukocyte values. And it is considered that propolis application would be beneficial to maintain the blood parameters before or during the cancer radiotherapy. The other point, propolis may reduce the organ damage and adverse effects induced by chemotherapy and radiotherapy treatments. Propolis reduces organ damage and warrants chemo or radiotherapy treatments in higher doses and longer periods. And of course, through this way, we can obtain higher efficacy through this way. Propolis and CAPE prevent cancer chemotherapeutics induce organ toxicity. This is also an important point. Unfortunately, all these data from animal experiments, for example, doxorubicin induce cardiotoxicity and neurotoxicity, cisplatin induce hepatic renal ototoxicity, methotrexate induce renal and hepatic toxicity, bleomycin-induced lung fibrosis inhibition and tamoxifen-induced liver toxicity may be inhibited or completely prevented by application of propolis and CAPE. Propolis and CAPE, is it beneficial in breast and uterine cancer treatment? This is also an animal experiment. CAPE, selective estrogen receptor modulator and binding to estrogen receptor beta, and prevent binding to estrogen receptor alpha in estrogen positive breast cancer cells, MCF7, MDA, MB 331. And CAPE did not increase uterine weight according to the result of this experiment. And CAPE, another important point, did not induce any risk for children for early puberty. Another point, this is also interesting. Professor Oğuz Öztürk and his colleagues, they, as I reported by the previous previous presentation by Alison, as you can see, the most important point here propolis and its flavonoid fraction on breast cancer cells were extremely higher comparing the effect of their effect, extremely higher comparing to the individual components, individual flavonoids. For example, they tested the effect of individual flavonoids, pinobanxin, quercetin, fumaric acid, but the combination of flavonoids were very highly, extremely comparing the individual components were active. This is one of the miracle provided, synergy miracle provided by the nature. Unfortunately, the number of clinical evidences in the international references not so high. mainly focused on oral mucositis treatment. Oral mucositis, in spite of high number of scientific evidences on the effect of propolis and cape on cancer, they were only clinically applied to reduce the symptoms, the complaints of oral mucositis as a frequent adverse effect during chemotherapy. So this is a meta-analysis study, head and neck cancers and breast cancer. The patients subjected to chemo or radiotherapy, and they tested the effect. They reviewed the effect of oral mucositis on these subjects. And among the 352 articles published on this subject, they selected 5 randomized controlled study covering 209 patients. And accordingly, propolis gargle may provide significant benefits in symptom control and treatment of complications of oral mucositis during chemotherapy or radiotherapy treatments. And also propolis increase iron absorption, thus may help to regulate the serum biochemical parameters. Yes. Except treatment of oral mucositis induced by chemotherapy or radiotherapy, the number of clinical evidences for the use of propolis in cancer therapy are rare. This presentation aims to draw attention of scientific community for future clinical opportunities which will be provided by propolis and cape in cancer therapy. I would like to thank you for your attention.
Sayın Profesör Yeşil'a da çok teşekkürler. Harika bir sunumdu. Propolis özelinde aslında doğal ürünlerin kanser ile ilişkili kanıtlarını bizlere sundunuz ve her bir kanser hücresi özelliğini nasıl belki de etkileyeceğiyle de ilgili özellikler de getirdiniz. Ben zamanı biraz aşmamak adına 2 soru da gelmişti. Onları da özetleyecek şekilde eğer bana bir sorunuz olmazsa...
Buyurun, onkolojist olan sizsiniz.
Teşekkür ederim. Çünkü klinik pratikte biz neredeyiz? Evet, çok farklı gelişmeler var. Kanser heterojen bir hastalık. Her bir organın kanseri çok farklı yolaklarla karşımıza çıkıyor. Ve kanser hücresi de kendi varlığını devam ettirmek için aslında sürekli saldırganlığını artıracak bir yol da buluyor. Moleküller değişik mutasyonlar vesaireyle. Şimdi propolisle ilgili çalışmalarda 2 farklı noktadan bakmak lazım belki. Bir prevensyon açısından, kanserin gelişimini önlemek açısından sizin ilk verdiğiniz Alman atasözüyle ilişkili aslında ve hem antienflamatuar etki hem antioksidan etki, immün sistemi modüle etmesi ve aynı zamanda tabii ki anjiyogenez üzerine de etkisiyle bir insanda kanser gelişmesini engelleyebilecek doğal bir ürün. Ve bununla ilgili yapılan çalışmalarda aslında net bir doz ya da protokol yok. Sorulardan biri buydu ama yine de tabii ki dediğim gibi bir kanserin gelişiminde genetik özellikler, çevresel özellikler ve çok farklı risk faktörleri de olduğu için ve hangi dozda, hangi süreyle kullanımı daha net belki bir katılımcımızın da yorumu vardı. İnsan araştırmaları hız kazanacak aslında bu kongreden sonra, ben buna inanıyorum. Çünkü fitoterapideki bazı çekincelerimiz aslında apiterapide yok. Biz klinik klinik pratikte, klinik onkolojide propolis kullanıyoruz. Sadece sizlerden bize sanıyorum daha yüksek kanıtla birlikte doz ve uygulama protokolü gerekiyor ve diğer arı ürünleri açısından da. Tabii kanser deyince diğer bir nokta propolis çalışması sizin de özetlediğiniz kanser tanısı almış bir hastada kanserin ilerlemesini durdurma. İşte o zaman biz kanser tedavisinden bahsediyoruz. Şu an hedefe yönelik tedaviler bir önceki İstanbul Üniversitesi'nden gelen sunumda her iki reseptörü üzerinden örnek verecek olursam hedefe yönelik antikorlar ve daha küçük moleküllü hücre içi ajanlar ve son zamanlarda da tabii son 7 yılın çığırı immünoterapi ile birlikte çok farklı aşamadayız. Propolis'in aslında bu bahsettiğiniz her bir kanser hücre özelliğini etkileme ve aynı zamanda immün modülasyonu da bir şekilde sağlaması ben yakın zamanda immünoterapi ile birlikte çalışmalarının da olacağını düşünüyorum. Geçen sene Türkiye'de arı zehri çalıştayında da bahsetmiştim immünoterapi ile birlikte çünkü radyasyon mesela immün sistemin kanser hücresine duyarlılığını artırıyor. Muhtemelen propolis de immünoterapi ile birlikte bu etkiyi gösterecektir. Biz nasıl farklı kemoterapötik ajanları eskiden kombine ediyorduk, nasıl şimdi immün kontrol nokta inhibitörleriyle birlikte diğer ajanları kombine ediyoruz. Çünkü dediğim gibi heterojen bir belki de bencil diyeceğimiz hücre var ve çok çok zeki yaşamak için. Tek bir noktadan vuramadığımız zaman farklı yerlerden bir şekilde hedeflemeye çalışıyoruz. Ben propolis'in tedavi ile ilişkili protokol dozu ve süresiyle ilgili de bir soru vardı. Net bir doz ve protokolü yok aslında şu an. Bizlerin de beklediği belki de bu. Ama kilograma 250 miligram gibi çalışmalar var ve CAPE içinde 500 miligram kilograma gibi çalışmalar var. Bunlar netleştikten sonra belki kanser tedavisindeki asıl kanıtlı yeri ortaya çıkacaktır. Fakat ben kısaca şu şekilde vurgulamak istiyorum. Klinik onkolojide biz propolis kullanıyoruz. Diğer arı ürünlerini de öneriyoruz. Özellikle bahsettiğiniz kemoterapi ilişkili yan etkilerin yönetimi mukozit dahil olmak üzere bunda kullanıyoruz. İmmün sistemin güçlenmesi için öneriyoruz. Aynı zamanda kanser ile ilişkili iştahsızlıkta bir şekilde öneriyoruz. Ve bunların bizim ilaçlarımızla herhangi bir ilaç etki azaltmasıyla ilgili bir olumsuz yeri olmadığı için de belki sinerjik etkisiyle de fayda görüyoruz. Bunun daha aydınlatılması gerekecek. Bir tek meme kanserinde hormon reseptör pozitif hastalarda, meme kanserli hastalarda özellikle etanol içerikli östrojen benzeri etki dair bazı çalışmalar vardı eskiden ama İstanbul Üniversitesi'nden gelen bu sunulan son çalışma ve sizin de bahsettiğiniz hem endometrium kanseri hem de meme kanserindeki yeriyle birlikte sanıyorum o çekince de biraz azalacak. Çünkü hormon pozitif, hormon reseptörü pozitif olan meme kanserli hastalarda biz propolisten biraz çekiniyorduk. Hani nasıl bir nane, tere gibi bazı bitkilerde de östrojen olduğunu biliyorsak propoliste de aynı benzer etki olabilir mi diye çekiniyorduk ama sanıyorum o da biraz klinik pratiğimizde değişiklik oluşturacak. Ben tekrar teşekkür ediyorum. Bunlar haricinde bana sormak istediğiniz çünkü çok güzel bir sunumdu. Benim
ben de bir şeyler ilave etmek istiyorum burada. Şimdi kanser kemoterapisinde, onkolojide uzun yıllar immünoterapiden kaçınıldı. Çünkü nedir? Kanser kemoterapisini olumsuz etkiler diye. Ama ne zamanki ilaç firmaları immünoterapi ile ilgili ürünler çıkardı, yani 200, 300, 400 bin, 500 binlik bir maliyete şey yapıyor. Birden immünoterapi ortaya yükseldi. Halbuki biz yıllardır söylüyoruz. Çünkü vücudun kendi immün sistemi, şurada belirttiğim gibi doğal katil hücreler tam katil, spesifik bir şey. Halbuki yıllardır bilim adamları sadece kanser hücresine ben nasıl bu kemoterapiyi gönderebilirim diye tepiniyor. Ama doğa bunu biliyor zaten. Anlatabiliyor muyum? Şimdi biz nerede, pozisyon olarak neredeyiz? Kuyruğunu yakalamaya çalışan kedi veya sıçan gibiyiz. Aynı yerde. Şimdi denenmediği için kullanılmıyor. Bilgi olmadığı için denenmiyor. Böyle fasit daire etrafında dönüyoruz. Önümüzde şey duruyor, kaynak duruyor ama şey yapmıyoruz. Şimdi mesela şu östrojenik kavramı bitkilerde, şimdi dediniz nane falan ama en kuvvetli östrojenler de fasulye, nohut. Değil mi? Fasulye yeme diyebiliyor musun insanlara? Çünkü bitkisel östrojenikler östrojen reseptör beta üzerinde etkili. Bu da kemikler için etkili. Mesela Japonlar hiç şey kullanmaz, süt ürünleri kullanmıyor ama kemikleri sağlam. Niye? Soya kullanıyorlar. Soya kullandıkları için ve kemikleri sağlam olmuş oluyor şey yaptığında. Bizde ise bir östrojen, aman östrojenik, aman östrojen, çocuğumun memesi çıkar mı, oğlum başka türlüye döner mi, cinsel tercih değişir mi, böyle bir şeyin içerisinde herkes. Yani öcü olarak östrojenik. Halbuki kersetin, soğan yiyor musunuz? Yiyorsunuz. Kersetin var, östrojenik. Elma yiyor musunuz? Yiyorsunuz. Yaban mersini yiyor musunuz? Yiyorsunuz. Hepsi östrojenik. Doğa östrojenik. Yani biz doğaya karşı çıkmaya çalışıyoruz. Mesela benim en nefret ettiğim ilaç grubu mide koruyucu diye satılan proton pompa inhibitörüdür. Sen doğaya karşı çıkıyorsun. Midenin asidini yok ediyorsun. Bu olacak şey mi ya? Mide asidi en kuvvetli dezenfektandır. Aldığın bütün mikroplar o zaman o olmazsa nereye gidiyor? Bağırsağa alt tarafa gidiyor. Bu defa patogenez ortaya çıkıyor. Yani burada bizim denememiz lazım, cesaret etmemiz lazım ve burada benim bu çalışmayı bayağı bana sıkıntı yarattı. Çünkü ben bir onkolog değilim, kanser çalışmış değilim. Anlatabiliyor muyum? Ama nasıl yaparım da insanların ilgisini buraya çekebilirim diye uğraştım ve benim çok zamanımı aldı bu. Beğendiğiniz için teşekkür ederim. Hani bir hata yaptıysam da affola. Çünkü ben neticede bir farmakognozi profesörüyüm. Yani etnofarmakoloji ile ilgileniyorum ama kanser ilgi alanımın dışında kalan bir şey. Hani diğer alanlarda şey yapıyorum. Bu bakımdan yani burada bir cesaret olması gerekiyor. Şimdi mesela propolis kullanıyoruz diyorsunuz ama hangi propolis? Çünkü hepsi nereye yönlendiriyor? Kape. Yani alkollü ekstrakta yöneliyor. Eğer kapasiz sulu ekstrak, çünkü bir de halkımızın nesi var? Aman alkollü olmasın, alkol değmesin bilmem ne. İlaçta herhangi bir o tip yaklaşım söz konusu değildir. Yani tükaka diye bir şey değildir. Çünkü Allah'ın bize emanet ettiği bir can var. Onu korumak bizim görevimiz. Etanol de neticede Allah tarafından yaratılmış bir şey. Onu alıp da kafayı dikip de alkollü şeyle sarhoş oluyorsan o zaman günah. İşte sapla samanı birbirinden insanlar ayırt edemiyorlar. Zaten bunun nedeni de kimler olduğu belli. Abuk sabuk şeyler, kehanetlerde bulunan insanlar habire televizyonlara, gazetelere çıkıyorlar. Oradan kaynaklanıyor. Yani burada denenmesi gerekiyor. Müthiş bir şey, koruyabilir. Siz güzel söylediniz, koruyucu olarak da kullanılabilir. Doz olarak hani doz yok deniyor ama şimdi mesela benim yayınlanmamış bir çalışma, yıllar önce kitabımı yazarken, apiterapi ile ilgili kitabı yazarken gördüydüm. Bir Japon doktor mide kanseri tedavisinde propolisi 35 gram dozda kullanmış. Yani çok yüksek ama bugün önerilen miktar genellikle 500 ila 1000 miligramdır ama bunlar da güvenilir doz nedir? Ona göre tespit etmek için yani bu biz eczacıların tek başına yapabileceği şey değil. Onkologlarla birlikte çalışılması lazım. Kolaborasyon lazım. Bu önemli bir şeydir. Yani birbirimizi görmezden gelemeyiz. Çünkü neticede her iki sağlık mesleği de insan sağlığı için yemin etmiş olanlar. Hipokrat yemini ediliyor. Nedir? Ben insana zarar vermeyeceğim önce. Ama kemoterapiyle insana zarar veriyorsun. Yani
Aslında tam öyle söylemeyelim. Hani kemoterapetiklerle hani çok başarı elde ettiğimiz dönemler oldu ama tabii bizim için onu hani yönetebilmek önemli. Çünkü her bir ilacın ya da her bir doğal maddelerin bile çok farklı şeyleri var. Ama hani şu dediğiniz şekilde özetleyecek olursak ve zamanı aşmamak adına çok keyifli bir toplantı. Sanıyorum dediğiniz gibi ortak çalışmaların kansere farklı açılardan, çünkü kanser multidisipliner bir hastalık. Tek bir branşın değil, tek bir temel tıbbın araştırması değil. Çok farklı bakış açılarıyla, çok farklı yaklaşımlarla aslında belki de bu noktaya geldik ve daha iyisine geleceğiz. Ben apiterapi açısından da dediğim gibi birazdan arı zehriyle ilgili bir oturum olacak. Orada da çok ilginç araştırma sonuçları var. Ve ben hani bunları bir şekilde farklı bir şekilde davetle tanıştım bu platformla ve oldukça mutluyum. Ufkumuzun genişleyeceğini ve çok farklı araştırmalarla aslında sizin de az önce vurguladığınız insana zarar vermeden insan için faydalı bir şekilde yol alacağımızı düşünüyorum. Ben tekrar organizasyon komitesine ve sizlere çok teşekkür ediyorum.